Hello. In this lecture, we're going to go over receptor signaling, especially the molecular pathways downstream from major type of receptors and how the signal integrated at the cellular level to perform different functions, as well as the dysregulation of receptor signaling will lead to different type of diseases. My name is Jian Li. I'm an assistant professor at Cell Biology and Anatomy at NYMC. Here is my email. If you have any questions about this lecture, feel free to reach out and contact me. All right. Before we start, I want to briefly review what you have learned from previous lecture on the signaling molecules that mediate cell-cell signaling. Generally speaking, there are two types of cell-cell signaling that are very important for multicellular organisms to communicate with each other and respond to environmental signals. This could be through direct cell-cell signaling. In other words, the molecule on the cell surface basically sends a signal to the neighboring cell. This could also through secreted molecules. Depending on the distance of the molecule that travels, we can characterize them into three major classes. If the signaling molecule go into the circulatory system, then that is endocrine signaling, and this is what this module will focus on. And if the signal molecule only travel a short distance and send a signal to the neighboring cell, this is called paracrine signaling. And if the signaling act on the cell, which is secreted the signal molecule itself, then that is called autocrine signaling. And in this lecture, we'll particularly focus on the endocrine signaling. The ligands or the signaling molecule in endocrine signaling also called hormones. And this particular contains peptides and lipid ligands that are secreted specifically from the endocrine cells and travel in the circulatory system and function at the distant site in the body. In Dr. Viswanathan's lecture, she already gave a very comprehensive overview of this hormone and its functions. I just want to basically remind you on two things. First, the peptide and lipid ligands have different biological properties. Peptides are usually polar and hydrophilic molecules. Because of this biochemical property, they are able to diffuse freely across the bilipid layer of the plasma membrane, so that they usually action on a cell-bound receptor and sends a signal through the receptor. On the other hand, the liposoluble chemicals such as steroid hormones, which can diffuse passively across plasma membrane, and therefore they can act with intracellular receptors. And in this lecture, we will basically describe the difference of these two types of receptors. I do want to remind you, in addition to peptide and lipid ligands, there are other type of signal molecules, including glycosylated proteins, carbohydrates, ions, and others. I want to start by introducing the general model of receptor signaling. In a receptor signaling pathway, usually it contains at least three components. First is the signal molecules, or the ligand, which recognize the receptor, which typically contains a complex protein or a tightly bound multipomer of proteins. The ligand binding to the receptor will introduce conformational change, and that will trigger the downstream effects. And that last part is called effectors, and introduce the changes in the cells and perform certain functions. The effectors could be activation of an ion channel that leads to the flow of ions across the plasma membrane, or different cellular compartments like ER or mitochondria, or it could be through a second messenger system, including the protein kinase, as we are going to introduce in this lecture. It's important to note each cell is programmed to respond to specific extracellular signal molecules due to the different expression level of different types of receptors. And this is the basis for animal development, tissue repair, immunity, as well as homeostasis. And the arrows in this signaling pathway analyze various human diseases such as cancer, autoimmunity, and diabetes. Generally speaking, there are two major types of cell receptors based on their location. The first is the cell memory receptors, and the second class is the intracellular receptors. Cell memory receptors contains several noticeable subset types. The first one is the ligand-gated ion channels, which the ligand will triggers the conformation change of the receptor and allow the flow of ions into the cells or outside the cells. The second type is called GPCR, G protein coupled receptor. And this is the type of receptors which mediated by a receptor bound G protein and introduce the downstream effector reactions. And third is the kinase link receptors. These type of receptors itself contains enzymatic activity. Upon binding of the ligand, this receptor will introduce a cascade of enzymatic reaction, therefore amplify the signal. These basically the three types of cell membrane receptors. An intracellular receptor are different from that because that receptor is not membrane bound or associated with the plasma membrane, but inside the cell. And these receptors could reside on different cellular compartments like the ER or mitochondria, but the most prominent one is the nuclear receptor family, which upon binding of the ligand could travel to the nuclear compartments and triggers a transcription factor activity. These different types of receptors have different time scale of actions. The gated ion channels and the GPCR really respond to the signal very quickly, and the kinase linked receptor and nuclear receptors will require a little bit longer time, usually hours, to trigger the downstream effect. And in the later slides, I will go through each of these classes and introduce to you the downstream signaling cascade. First, let's go over the ion channel linked receptors. This is a type of receptor frequently found for the neurotransmitter signal at the synapses. The receptor typically contains at least two different important parts. One is the ligand binding site, which will receive the signal from the extracellular matrix and extracellular space. And the second part is the ion channel core that upon binding of the ligand will cause the conformation change, open up the core, and allow the ion to flow through the plasma membrane. When that occurs at the synapses, the presynaptic neurons secrete the neurotransmitter, which is the ligand, which recognize the receptor, recognized by the receptors at the postsynaptic neurons, and open up the channel and allow the ion flow. If this results in a depolarization of the postsynaptic neurons, usually that has a excitationary role. And if this lead to a hyperpolarization, and that will inhibit the neuron response of the postsynaptic neurons. And during this process, you can imagine the chemical signals from the presynaptic neurons will be converted into an electrical signal in the postsynaptic neurons. The second type of membrane-bound nuclear receptor is called G-protein coupled receptor. This actually is a very big class of receptors. And noticeably, this is also a very important drug target. By far, about more than 1/3 of FDA-approved drugs actually target GPCR and its related pathway. At the biochemical level, GPCR is a 7-pass transmembrane protein, which is very unique, and we will come back to this feature later. And it contains extracellular ligand binding site. And in the intracellular side, they are associated with G protein, which contains 3 subunits, the G alpha, G beta, and gamma. In the inactive state, G alpha is associated tightly with G beta and gamma, and upon activation by the extracellular signal or by binding to the ligand, G alpha dissociate with G beta and gamma. This will allow G alpha associated with GTP. That will activate the G alpha. Also, the dissociation will activate G beta and gamma as well. For the sake of time, in this lecture, we'll particularly focus on the effectors that's associated with G alpha. But I do want to mention G beta and gamma could also function through their own effector and initiate signal transmission. And the G alpha, when it associated with GTP, is in the active state. If you transmit the signal through its downstream effectors, noticeably, the G alpha itself contains the GTPase activity, which means if you hydrolyze GTP into GTP, that is the inactive state. In that state, G alpha will reassociate with G beta and gamma and return to the inactive state. So, because of this GTPase activity, it will form a feedback regulation on its own that will determine the duration of that active state of G alpha. We'll come back to this point later in this lecture. There are 2 prominent or primary effector pathway associated with the G alpha. One is the cyclin AMP pathway. Another one is the IP3 and DAG pathway. So let's look at the CMP pathway first. This is through an action of G alpha with the adenylate cyclase, or AC. So activated G alpha could either stimulate the activity of AC or inhibit the activity of AC, depends on the type of G-protein coupled receptor and depends on the ligand. For example, for the stimulated G alpha, they are associated with certain types of the GPCR, which will activate the adenylate cyclase that will stimulate the production of CMP. But on the other hand, there are also inhibitory G protein alpha that associate different types of GPCR, like in this figure shows the dopamine receptor, and that would inhibit the activity of AC and CMP activity. CMP serves as a secondary messenger. It could activate the later effector, including the protease kinase Ka, as a pKa as a very prominent example, and introduce the protein kinase cascade and the signal transduction. On the other hand, the other primary effector of GPCR for example, as the downstream signal for the Q-type of G-alpha protein could be the phospholipid C that associates the plasma membrane. These could generate a two important second messenger through the membrane bound phosphoadienosyl 4,5 biphosphate. The cleavage of the substrate could generate the inosine 1,4,5 triphosphate IP3 as well as the diacetylglycerol DAG. These serve as important second messenger and induce the activity of protein kinase C as well as the release of calcium and downstream signal transduction. We'll take a closer look at these two downstream signal transduction pathway in these two slides. First, the PPKA signaling pathway. As I mentioned, certain type of GPCR could associate with the stimulatory G protein that could activate the AC and CMP production. There are also inhibitory G protein type of GPCR that could inhibit AC as well as CMP. CMP as an important secondary passenger could introduce the conformation change of PKA. The PKA contains the regulatory subunit as well as a catalytic subunit. In the innate state, they are associated with each other, and the regulatory subunit will inhibit catalytic activity of PKA. Upon binding to CMP, will release the regulatory subunit from the catalytic subunit, release the activated PKA. These PKA can function on different substrates in the cytosol as well as in the nuclei, including transcription factor CRAB as shown in this diagram, and induced downstream transcription reaction. PKA is a very important serine and threonine specific protein kinase, and is a very important secondary factor in this pathway, and regulates cellular functions through various targets. On the other hand, the IP3 and DAG pathway use different mechanism. These usually function on the Q type of G protein, and this is triggered by the activity of PLC on the plasma membrane. Use the PIP2 as a substrate. You can basically produce IP3 as well as DAG as the product. IP3 can bind to the IP3 receptor on the surface of ER endoplasmic reticulum or on the membrane of mitochondria. This will trigger the release of calcium from the ER or the mitochondria. And the elevated calcium also allostatically activates protein, including the calmodulin and activated calmodulin kinase pathway that triggers, different cellular response with the kinase cascade. On the other hand, the elevated calcium could also activate many protein kinase C forms, and this will convert the signal with the DAG, which also activated protein kinase C, and then amplify the signal and triggers the PKC pathway. The third type of cell membrane receptor is called enzyme-linked receptor. Different from the G protein coupled receptor, this receptor itself have the kinase activity and depends on the specificity of the kinase on different residues. They can be categorized with different subtypes. The most prominent subtype is the receptor tyrosine kinase, meaning the kinase activity of the receptor will trigger phosphorylation on the tyrosine residues. Here is a example of the receptor tyrosine kinase. Uh, upon binding of the ligand, uh, tyrosine receptor tyrosine kinase dimerize and activate itself by autophosphorylation. And this autophosphorylation on the right tyrosine residue in the intracellular phase will attract the adapter protein. In this case, it will activate an intermediate protein called GNRP, which activates the ROS G protein. The ROS G protein therefore recruits RAF to the plasma membrane and then activate it. This will trigger the very important kinase pathway called MAP kinase pathway, and then induce downstream kinase cascade as well as transcription response. On the other hand, the tyrosine kinase associated with the receptor would also trap a protein called GAP, which is a GTPase activating protein, right? This would function on the RAS G protein and return the G protein from the active GTP bound phase to the inactive GDP phase, and then turn off this pathway. This is similar as the case I introduced with GPCR pathway, but instead of using the G protein itself as feedback regulation, you use the adapter protein GAP. But nevertheless, the similar feedback mechanism is turned on, and then that controls the strength as well as the duration of the receptor tyrosine kinase activation. Another very noticeable example of the receptor tyrosine kinase is the insulin or insulin-like growth factor signaling pathway. They're very similar, the ligand very similar at the biochemical level, and the functions on the insulin receptor or the insulin growth factor receptor, and triggers similar downstream pathway. First is it triggers the MAP kinase pathway, similarly as what I introduced in the previous slide. That's through the ROS as well as the MAP kinase, and then this branch of the insulin IGF signaling pathway controls the cell growth as well as differentiation. On the other hand, the insulin signaling pathway can also recruit, upon activation, can recruit another effector called SHP2. This would activate a different kinase pathway called PI3K AKT pathway. This pathway would further activate a very prominent kinase called mTOR. This is a famous kinase complex which contains protein homeostasis. It activates protein translation as well as suppressed autophagy upon activation, so that it controls the protein synthesis and the degradation balance. On the other hand, AKT also controls the phosphorylation of a bunch of important transcription factors. The most prominent one could be the FOXO transcription factor, which controls metabolic stress response. Overall, this pathway controls the metabolic action of the insulin IGF signaling pathway. A third receptor tyrosine kinase example I want to give is the JAK-STAT pathway. The importance of this is revealed by research in the mammary gland development as well as the milk production. So in this case, the percolin receptor responds to ligand prolactin, and upon dimerization activation, the JAK kinase is activated. It functions through the downstream transcription factor called STAT5. In the inert state, STAT5 is present as the monomer and is inactive. And upon activation of JAK2 kinase, it phosphorylates step 5 at a certain tyrosine residue that will trigger dimerization step 5. Step 5 therefore translocated into the nuclei and triggers transcription activity and activates the expression of the downstream genes, including a very important gene called casein that is involved in milk production. A similar pathway that through the receptor tyrosine kinase that can activate STAT is the FGF receptor, fibroblast growth factor receptor 3. These, using very similar mechanism, turn on the STAT1 transcription factor. And this is very important or essential for the human fetal bone growth. When there is a mutation, loss of function mutation of this kinase, or a premature activation of this kinase, it will disrupt the balance of the STAT1 transcription program, lead to cartilage growth stop before birth. That will lead to very narrow chest, extreme short limbs, and then cause severe developmental disorders. In addition to the receptor tyrosine kinase, there is also other enzyme-linked receptor. One of the prominent example is the receptor serine or serine kinase. This kinase use very similar mechanism for activation. However, the activity of the kinase largely on the serine or serine residue of the substrate. One of the prominent example of this type of enzyme-linked receptor is TGF-beta receptor. Different from the insulin receptor, this TGF-beta receptor contains both type 1 as well as type 2. It's not like the insulin pathway, they have the hormone dimer formation. In this case, they have a heterodimer formation upon activation or upon binding to the TGF-beta like ligands. That will triggers the kinase activity. The downstream effector of this signal pathway is the SMAD family of transcription factors. Upon phosphorylation, they will form the heterodimer of SMAD, either through SMAD 2, 3, and 4, or 1, 5, and 4, that will triggers the downstream transcription activity, and then alter the transcription program. Finally, I want to go to the last type of receptor that's very different from the receptor I already introduced. That is the intracellular receptor. As I mentioned, there are receptors on different cellular compartments like ER and mitochondria, but the most prominent example of this subtype is the nuclear receptor. Here, I want to basically use the glucocorticoid receptor as an example. This, of course, as you guys know, have very important functions in immune response. A general theme of this type of nuclear receptor is they are associated with molecular chaperones like the He-shell protein in the inert state. Upon binding of the steroid, the He-shell protein is released from the receptor. That will allow the nuclear localization of this receptor and convert that into an active transcription factor. In this case, the glucocorticoid receptor will recruit a co-activator or co-repressor depending on the context and then alter the transcription output. Similar nuclear receptor, including the estrogen receptor, progesterone receptor, as well as the androgen receptor, which have a very prominent role in reproduction and then gametogenesis. Okay, here I think I would like to quickly review the major receptor classes I introduced to you and on the difference of their effector and the coupling mechanism as well as their biochemical properties. Okay, so we introduced the three membrane bound receptors, the G protein coupled receptor GPCR. These use the G protein for the coupling mechanism, and it, it contains 7 transmembrane helixes for the receptor, and I will come back to this point in a later slide and review the clinical relevance of this type of receptor when mutation occurs. On the other hand, the ligand-gated ion channels doesn't really need an intermediate protein to introduce the downstream effect. It could have a direct effect, upon binding of the ligand, could allow the flow of ions into the cell or outside the cell, then have the downstream effect. The receptor kinase also could have a direct effect, but through the intracellular kinase domain, introduce the kinase cascade. A prominent example of this including insulin, growth factor, as well as the cytokine receptors. This receptor only contains a single transmembrane helix that link the intracellular receptor part that receive the signal and bind to ligand and the intracellular kinase domain. Different from this membrane bound receptor, nuclear receptor doesn't reside on the plasma membrane, but located inside the cell. And a nuclear receptor, upon binding to ligand, could convert into a transcription factor that recruits either co-activator or co-repressor and change the transcription program. Here is the cartoon, basically go over these different types of receptors and its downstream pathway. For the membrane bound receptor, usually the associated with the hydrophilic signal molecule, which cannot freely pass through the membrane, so that you need this membrane bound receptor to receive the signal and transduce the signal inside the cell. GPCR use the major effector. There are 2. One is the adenine cyclase, which produce CMP. Depends on type of GPCR. It could be either positively regulates activity of AC or negatively regulates activity of AC, then change the CMP that will further change the downstream kinase, including pKa. The other prominent effector of GPCR is through IP3 and DAG to regulate the protein kinase C activity and the downstream effectors. For ligand-gated ion channel, the binding will introduce ion flow into the cell, and for the receptor kinase, in this diagram shows the receptor tyrosine kinase as a prominent example. This will triggers the MAP kinase pathway through the G protein RAS. I do also want to mention, in addition to this receptor signaling pathway I introduced that binds to either the protein or lipid for signal transduction. Inside the cell, there are other signal molecules. In this diagram, shows the nitric oxide and carbon monoxide, which also serve as a neurotransmitter, which could through different type of receptor that triggers the instead of CAMP, but the CGMP, and then activate the protein kinase G. All right. So I also want to take the chance to review the features of receptor signaling as I introduced in my earlier slides. That means there are a couple of the important common features of the receptor signaling. First, that is signal integration. The cells, based on the expression of different types of receptor, would integrate response and integrates different signals from outside. And the second feature is signal amplification through different effectors which could converge at certain point of the signal transduction pathway. The signal would get amplified. Third is the feedback regulation, which is very important because you do not like the receptor always keep at either the inactive or active state, so that a feedback regulation is needed to regulate the duration and the strength of the signaling. Okay, here I would like to use the GPCR and CMP pathway as example to illustrate these three features of signal transduction. As I already mentioned, the GPCR could have either associated with a stimulatory G-alpha subunit or the inhibitory G-alpha subunit, depends on the type of the receptor as well as the ligand. In this case, adrenaline work on this stimulatory GPCR, which will stimulate the production of CMP. On the other hand, acetylcholine could function with inhibitory GPCR and inhibit production of CMP. Through the downstream protein kinase A, it would influence the cellular activity. That is a part of signal integration. pKa, as a very important kinase, have very many substrates, including the lipase and glycogen synthesis, as well as the phosphorylase kinase. Through the different targets, I hope you would appreciate if you change the metabolic program. For example, the lipase will, upon phosphorylation, will increase lipolysis. And for the glycogen synthesis, upon phosphorylation, that will reduce the glycogen synthesis. On the other hand, the phosphorylase and phosphorylase kinase would actually increase glycogen breakdown. You could appreciate from here that the overall net effect of those pKa substrate functioning in the metabolic pathway that will increase the catabolism, that break down the lipid as well as break down the glycogen to produce glucose. This through a concerted function that either reduce the glycogen synthesis, in the meantime, increase the breakdown of the glycogen. This is a part of a function that the signal transduction pathway could amplify the signal and have a strong cellular function on the metabolism in this case. Finally, I want to also remind you that the G-alpha subunit of G protein have the intrinsic GTPase activity, so that they provide a natural feedback mechanism so that you will not keep this GPCR always at active state. Upon this hydrolysis of GTP to GTP, G-alpha protein will return to the inactive state, therefore regulates the duration of the activation state. Finally, I want to basically spend the last few slides to talk about dysregulation receptor signaling and its implication in disease. First, I want to mention the dysregulation of receptor signaling could function on both the loss function or gain of function side. All right, here I just show you some example of loss of function of GPCR. This diagram labels the mutations that could either inactivate the receptor or cause misfolding or trafficking defect of this receptor. In both cases, it will reduce the function of GPCR. Shown you here are two very important receptor to gonadotropic hormone, FHR and LHCGR. Okay, the right color labeled mutation are the mutations that inactivate the activity of receptor. And the yellow one, you would appreciate that probably have equal number or even more number usually reside in the helix region that form the transmembrane domain of GPCR. And those mutations that naturally contact the ligand were associated with G protein, yet will still cause loss of function effect. And those mutations have this effect through the structure change of the receptor. Because this unique structure of the 7 transmembrane domain, this protein is very sensitive to protein folding as well as trafficking because they have to go through the Golgi pathway to reach the plasma membrane. And those mutations will disrupt either the protein folding or the trafficking. Therefore, lead to this receptor reduced on the plasma membrane, therefore loss of function. On the other hand, there are also gain of function mutations in the receptor signaling pathway. Here, I want to show you one example of estrogen receptor alpha mutation, that the Y537S mutation. This mutation is a gain of function mutation. First, this one will keep the ER alpha at constitutively active state. In other words, it doesn't really require the binding to the ligand estrogen to keep it active. Second, these mutations also has a very bad feature that is insensitive to the endocrine therapy. In the wild-type ER alpha case, if we apply the selective estrogen receptor modulators, this is a type of drug could competitively bind to the ER alpha and then disrupt its transcription activity and or lead to its degradation. However, in this mutant case, the mutant protein is insensitive to treatment of these selective estrogen receptor modulators, so that it will always keep the transcription activated on its target genes. Therefore, this will underline the very aggressive subtypes of breast cancer. Because of the importance of cellular function and the clinical relevance, there are a lot of drugs that have been developed targeting the receptor signaling pathway in therapeutic. Here, I only show you a very short list of FDA-approved drugs that targets some of the receptor signaling pathway that were introduced in cancer. First, this drug that targets the AKT, that downstream of the insulin PI3K pathway and related to also upstream from the mTOR pathway. This has been approved for the treatment of breast cancer. And this is the drug that is target the ER alpha. This drug serves as a selective estrogen receptor degrader. In other words, it will cause the degradation of ER alpha so that shut off the ER alpha pathway in breast cancer. And the last drug I want to highlight is that very interesting. It is specifically designed for a very aggressive cancer type that caused by KRAS, the RAS protein that initiates the MAP kinase cascade. The specific mutant is the G12C mutation. This is a mutation that will keep KRAS constitutively active. And the drug is developed to switch this constitutively active mutant off. therefore shut off the tyrosine receptor kinase as well as the downstream MAP kinase pathway that stimulate cell proliferation in cancer. I hope with this lecture, you will have a feeling of the general mechanism of receptor signaling pathway and its relevance for disease as well as the drug development. And take this knowledge to learn other signaling pathways that I couldn't include in this short lecture. And as I said, if you have questions, feel free to reach me through the email. Thank you.